Good morning. Thank you for the invitation, Scott and Marie. I hope that some of this information is actually useful. I think this is probably trying to make a case for metabolomics in general. A great deal of emphasis has been placed in taxonomic identification of biomarkers on different conditions, and this study really indicates the fact that a lot of bacteria can do a lot of things, and so under different set of conditions, a proteolytic bacterium could be doing some carbohydrate breakdown. So I think that we're looking for biomarkers for looking at taxonomy might be a little difficult based upon the prolific metabolic capabilities of bacteria. And even at the strain level, there are differences that actually take place in the metabolic profile. We looked, we used the model as a metabolic flux model in which we assembled about 350 different IBD strains, different metagenomes, and then we used that as a basis and introduced a single additional organism into that metagenome and look at the fluxes in the metabolic profiles, and you can see that not only there are shifts in the metabolome, but also, at least for individual metabolite, but also you can see that all the different strains have different impacts on metabolomes. Ergo, so the microbiome studies really that are focused specifically solely on taxonomy are not going to give you the information that you're asking for, which is actionable information. So therefore, while taxonomy is wonderful and easy to sometimes obtain, metabolomics is going to have to play a key role in identifying biomarkers for health and disease and ultimately define the elusive healthy microbiome. So the goals of the Metabolome Reference Standard is to develop a human gut microbiome reference material prepared from human stools. To promote the adoption, hopefully, presumably, of the reference material by a scientific community to validate current protocols and benchmark them. So it is anticipated that should all this come to fruition, we'll make all the data generated from this material available in an Amazon S3 bucket somewhere in cyberspace so the user can access it and look for comparing the protocols. And at some point, this is the big hope, and it's very likely that it's never going to happen, but it would be nice to believe that it's the adoption of journals, the golden standards and a requirement really before they can publish any meaningful data. So the implementation of the project is, the Biocollective prepared the samples for the Mosaic Challenge. And during that time, we learned that we can prepare large quantities of fecal samples in a homogeneous and stable manner. So the Biocollective will develop the material. Already we have some material in inventory, and it's undergoing homogeneity and stability studies at the microbiome, the taxonomic, both 16S and shotgun, and NIST is conducting the corresponding metabolomic standard stability. The reference material is collected from whole stools. We may collect several kilograms, really, of whole stools from different donors, and we can either identify from our own database them being healthy, self-declared healthy, or with inflammatory bowel disease, or with Parkinson's disease, we have several different cohorts from which we can reach out for donations. And the processing of the fecal sample, we argued about whether I should, break them all into little states. But the idea is to collect them, test them for pathogenicity or lack thereof, and we screen them for all enteric pathogens, HCV, HBV, HIV. So and once we determine that this particular fecal sample is not carrying significant pathogens, then we'll pull them together, we homogenize them in bulk, and this bulk then is quantified and aliquoted into different volumes, anywhere between 1 gram, which is a lot of stuff, to 100 milligrams. And since we are going to have to aliquot them, the amount that we prepare, the smallest quantity, in other words, we shoot for 100 milligrams per milliliter, so that we can actually process the fecal sample by filtering, taking the tomato seeds and all the fibers and all of the clumps that might be in there, not necessarily bowling balls, but pretty close. And at that time, then we are able to aliquot it to increase the propensity for homogeneous product. In this particular case, we do not spike it. For the Mosaic challenge, we did spike it with 10 to the 8, and that in itself presented its own challenges. So we prepare these samples. This particular set of samples that we have now have come from self-declared healthy, proceeding from about 3 kilograms of human feces, 4 different donors, 5 different donors, 4 different sequential samplings, all mixed. So we were shooting for diversity rather than identification. In fact, that was one of the things that we required, that we cannot trace back the identity even by deep sequencing, because it makes it very difficult when you have 5 different stools from 5 different donors all mixed together. So we sent some of these samples to Zymo. It was a random decision, and we sent them 5 aliquots, and these aliquots were processed using their proprietary DNA, RNA later, and they're using just phosphate glycerol, which is a common storage material for fecal samples. And while the samples are shown to be homogeneous, it is obvious that different sample preparations and sample processing will give you slightly or significantly different results. However, these were tested statistically. The intra-sample variability is not significantly different. So all the individual aliquots of a given sample are shown to be statistically not significant in different composition. And in fact, the level of homogeneity is greater than really initially expected. And then we then took another set of aliquots and processed it using the Cosmos ID, primarily pipeline, just simply because we wanted a different vendor, and we want to have nothing to do with measuring the homogeneity of the product. And what we did was, we used different concentrations of dry mass, and also different methods of preservation. The samples, Scott will tell you the samples that we shared with NIST, but in generally speaking, broadly speaking, the concentration of the amount of material that is included it allows you for some degree of inhomogeneity among the different groups, and the reason primarily is because at 250 milligrams per milliliter, some stool samples are very difficult to homogenize. They become the blob. So we found that the optimal concentration is 100 milligrams per milliliter, and that is what our product is like. So looking at sample 1 alone, you can see that, or that's glycerol and 100 milligrams per milliliter sample. There is some inhomogeneity, but most of which the last 2 samples are reflecting because they were lyophilized rather than in phosphate buffer saline. But I do not know whether the 2 of them are correct or incorrect, but regardless, there is homogeneity in the product. We also, we are trying to ask a question, from a metagenomic point of view, which are the taxa that I am going to look for the stability studies. And I opted for the core microbiome just as a first pass at 100%. So we see this taxa showing up at hopefully approximately the same concentration in each of the samples, in all of the samples. And this is a list of some of the core microbiome. It's probably a little longer, and that's going to be part of the product information sheet that we will provide. The data, the prior data was 16S ribosomal RNA. This is shotgun data. And so we are comparing the taxonomic profile with a relatively limited metabolic profile of looking for just virulence factors identified in the database, identified by searching the virulence factor database. And again, the point that I'm trying to make is while stability studies are under development, or we are at the 6-month stage right now, the homogeneity studies, at least in our estimation, the samples are satisfactory.